In this lecture, we're going to talk about the citric acid cycle, which I'm sure you've been taught before ad nauseum, but the reason why you'll see it over and over again in your courses is because it really is the metabolic hub of the cell. All of the fuel molecules are going to funnel into this cycle and be oxidized so that we can make a huge amount of ATP. Acetyl-CoA is going to be the starting molecule used in the citric acid cycle, which also goes by a couple different versions of abbreviations, or CAC for short. It also goes by the name of the tricarboxylic acid cycle, or TCA, as well as the Krebs cycle after Hans Krebs, who discovered the reactions involved in this pathway. Now, I'm sure you've covered it a billion times in your biology courses, but we are going to focus on just how important it is as a central pathway in metabolism and energy production. We'll look at exactly what happens as molecules move through the cycle, and we'll also see similar trends in terms of regulation patterns, where high energy and other intermediate metabolites can slow down the various steps. These supplemental readings will take you through the cycle step by step, and we will walk through them, but make sure you focus on how the pathway is regulated and the high energy intermediates that are made. Now, I saw this on Imgur and it reminded me of the metabolic pathways. There's sometimes so much going on, it's hard to keep track of where molecules are going. And it's really going to be the regulation that's going to be so very important for a cell to make what it needs. Just coming back to my cartoon summary of the main pathways, acetyl-CoA is going to enter into the citric acid cycle, and this acetyl-CoA can come from either the breakdown of glucose to pyruvate in glycolysis, and then the pyruvate dehydrogenase complex, which we saw in the last lecture, or it can come from amino acids, as well as the breakdown of fats in a process called beta oxidation, which we'll look at a little later on in the course. We're not really going to look at the urea cycle, but this is how your body gets rid of ammonia or the nitrogen from amino acids. And there are links between the pathways. And you can actually also make some of the amino acids from the citric acid cycle intermediates. So the citric acid cycle really is the second stage of cellular respiration, where those macromolecules are going to be broken down to produce the amphibolic intermediate acetyl-CoA, as well as other citric acid cycle intermediates, which can either get oxidized to produce ATP and CO2, or can get used for fat or even glucose synthesis. So we saw in the last lecture how pyruvate can get converted to acetyl-CoA, and I mentioned other fuel molecules as well, such as fat, amino acids can also be converted to acetyl-CoA. And acetyl-CoA is going to be the starting molecule coming into the citric acid cycle. And we'll see that the electrons from the intermediates in the cycle will actually be used to reduce NAD+ and FAD in order to make ATP later on in oxidative phosphorylation. So these 8 electrons in this figure are really the reduced coenzymes NADH and FADH2, and they're going to get used in the electron transport chain in order to make ATP. That's one of the main goals of the citric acid cycle, to oxidize molecules to carbon dioxide and temporarily hold the energy of oxidation in the reduced electron carriers NADH and FADH2. We'll see later on their role in oxidative phosphorylation and the generation of ATP in the mitochondria. So this is the overview of the cycle, and there are 8 reactions in the citric acid cycle where the enzymes are shown in, their names are shown in blue. And we will actually start at the top left with acetyl-CoA from the breakdown of carbs, fats, or proteins. The citric acid cycle is usually broken down into 2 stages. The first 4 enzyme catalyzed reactions would be citrate synthase, aconitase, isocitrate dehydrogenase, and alpha-ketoglutarate dehydrogenase complex will all be found in stage 1. And then the last 4 enzymes are going to be in stage 2. So stage 1 is going to include the 2 oxidative decarboxylation steps, where we're actually going to make 2 CO2 molecules as well as NADHs. And in the second half of stage 2, it'll actually involve the production of a GTP in mammals. And this GTP is made in a process called substrate level phosphorylation, since we are phosphorylating GDP to GTP. And then we're also going to make another reduced NADH and an FADH2. We're going to walk through the cycle and answer a few questions so that you can understand what's going on. The first is, why is the citric acid cycle considered a cycle and not a linear pathway? When we start with this green acetyl-CoA molecule, it actually joins with a molecule called oxaloacetate. And this is actually the final product of the cycle. So this is why it is called a cycle. The pathway is dependent on the final product in order for that first reaction to occur. So oxaloacetate and acetyl-CoA are going to join, and this will all be catalyzed by an enzyme called citrate synthase. And oxaloacetate is actually a limiting factor, okay? So if you don't have a lot of oxaloacetate present, the cycle won't be running all that fast, okay? It won't be able to use up all of the acetyl-CoA in order to generate energy. And when we combine the acetyl-CoA with that oxaloacetate, we're actually going to be using the presence of that thioester bond, okay? So we're going to be hydrolyzing it, and that's what's going to join these two molecules together. Okay, so it's actually creating a larger molecule. So let's just look at that reaction. When acetyl-CoA enters the citric acid cycle, we are going to couple the hydrolysis of that thioester bond with the joining of the methyl carbon on the acetyl group to the carbonyl at C2 of oxaloacetate. So even though water is involved in this reaction, and it is technically hydrolyzing off that bond, the enzyme's actually classified as a transferase, since we're transferring that CH2COO- group from coenzyme A onto oxaloacetate. Now the standard free energy change for this net reaction is negative, and favorable is written in the forward direction. And this is the net reaction, so it takes into account all of that energy that's released when the thioester bond is broken, releasing free coenzyme A, but also joining oxaloacetate and that methyl carbon. That coenzyme A could then get used by other reactions, such as the pyruvate dehydrogenase complex for its reaction. So now, if you look at the molecule that's made, the biochemist in you really should recognize that it is indeed citrate that's made, not citric acid, as the name of the cycle suggests. So remember your pKa values? The carboxylic acid groups actually have low pKa values. So at physiological pH, these groups are going to be deprotonated to form the conjugate base. Now there's another name for the cycle, the tricarboxylic acid cycle, and it's also partially incorrect since it does come from this molecule. And even though we do see 3 carboxylic acid groups, so that would be the tri part on citrate, they really aren't acids but rather the conjugate base. So just to summarize, when acetyl-CoA comes in, we are joining it with the final product of the cycle, oxaloacetate, in a favorable reaction. All right, so why is the citric acid cycle considered a cycle? It's because it's dependent on the final product, oxaloacetate. Let's look at the next question. What is the net reaction for the citric acid cycle? So just building on question 1, since oxaloacetate is used as a substrate, but it's also produced in the last reaction, it's actually not going to be included in the net reaction. So let's go back to the cycle and look at what molecules are required and what is made. So we'll start with, obviously, acetyl-CoA. So we're going to start with the cycle, and it's going to need an acetyl-CoA as a starting substrate, as well as a water molecule for this first step. And there's actually another water molecule that's going to be required by fumarase down here. So there's actually going to be 2 waters in total required as substrates. We're also going to produce 2 carbon dioxide molecules in stage 1. So that's going to be down here at step 3 and 4. And we're also going to be making 2 NADHs there, as well as another NADH by malate dehydrogenase. So a total of 3 NADHs. Now, before we look at anything else that's made, I just want to talk a bit about some of the enzymes that are present. So citrate synthase we saw was a transferase where we were moving the CH2 and COO- group onto oxaloacetate. Step number 2, aconitase, is just a rearrangement, so an isomerase where the OH group gets moved onto this carbon. And even though steps 3 and 4 are both redox reactions or dehydrogenases, they actually have very different mechanisms of action. If we look at the redox reaction catalyzed by isocitrate dehydrogenase, We'll first see that there's a reduction of NAD plus to NADH by a hydride. Okay, and along with this, because it is a redox reaction, we have the pairs where we'll also see the oxidation of the alcohol to a carbonyl, and that's going to produce this oxalosuccinate intermediate. We'll then see the decarboxylation reaction where we're going to lose this blue COO minus group to produce alpha ketoglutarate. Now, this final product, alpha ketoglutarate, this molecule should sound a bit familiar, even look a little bit familiar to you, because it's actually just glutamate, that amino acid that we learned about, where we've got a carbonyl instead of an amine on this alpha carbon. So this is just one way you can get amino acids into the citric acid cycle. If we compare isocitrate dehydrogenase to the next redox reaction, alpha ketoglutarate dehydrogenase complex, this enzyme is actually a complex of enzymes and very similar to the pyruvate dehydrogenase complex where we saw that CO2 was first released and then the NADH was made. Okay, so alpha ketoglutarate dehydrogenase also has 3 different subunits where E1 is first going to catalyze the decarboxylation reaction that we saw similarly in the pyruvate dehydrogenase complex, and then NADH will be made by E3. We're also going to be adding on coenzyme A to form succinyl-CoA, and this new thioester bond will help drive the next reaction in the cycle. Now, what's really cool is that these enzymes are actually a great example of the evolution of enzymes, where different substrate specificities can arise using the same mechanism of action. Okay, so if you compare the substrates, we've got lots of similarities up here that are just highlighted in orange. We just have an extra methyl and COO minus group. So that would indicate to you that there might be slight changes in the amino acid side chains that are present in those active sites. So just coming back to the pathway in order to finish off our net reaction. In stage 2, we're also going to make a GTP in that synthetase reaction using the thioester bond. And then we'll have 2 more redox reactions producing FADH2 and then that third NADH that I mentioned before. So in order to round out the entire net equation, obviously we're going to require an inorganic phosphate and a GDP substrates in order to make GTP. And then you'll also see down here that CoA is going to get used in a reaction and produced as well. So really the net reaction just produces one coenzyme A. So here's the net reaction. We start with acetyl-CoA and we also require 2 water molecules in 2 of the steps. We'll be making 1 GTP from a GDP and an inorganic phosphate. And we're also going to make 3 NADHs from 3 NAD pluses, an FADH2 from an FAD, and then we'll also produce 2 CO2 molecules as well as releasing that 1 coenzyme A. Now, the last thing we need to talk about is where the CO2 carbons originate, and this is actually color-coded in the cycle. So if you look at the carbons on acetyl-CoA, they're actually color-coded in green. And if you follow them through the intermediates of the citric acid cycle, you should see that they actually stay on the intermediates. Okay, the CO2 molecules that are released are actually color-coded in black. So this means that those carbons actually originated from oxaloacetate. Now, studies were actually done in the lab where they used radiolabeled carbon in glucose in order to track the movement of carbons through the pathway. Now, if we keep following the green carbons, we get to a point in the pathway where the succinate molecule is symmetric. So we can't actually tell if the top 2 carbons came from acetyl-CoA or the bottom 2. So I know they're all pictured in black, but it's kind of like a hybrid between black and green. We don't really know. So in the first round of the cycle, we do lose 2 CO2 molecules, but those carbons don't originate from acetyl-CoA, they come from oxaloacetate. In the next round of the cycle, there's actually a 50% chance that the CO2 molecules did come from that first acetyl-CoA. So even though oxaloacetate isn't found in the net equation, it is a very important molecule. It's the key starting substrate and the donor for the decarboxylation steps, and its production is one of the goals of the second half of the citric acid cycle, so stage 2. Okay, really regenerating oxaloacetate in order for the cycle to run again. So these really are the main points for the citric acid cycle. Okay, we can break it down into 2 stages where acetyl-CoA comes in and reacts with oxaloacetate to form citrate, and there will be 2 oxidative decarboxylation steps to produce 2 CO2 molecules, where the carbons came originally from oxaloacetate, not acetyl-CoA. We then have the production of 2 NADHs, and then in the second half of the pathway, we'll regenerate oxaloacetate as well as producing 1 FADH2 and another NADH as well as a GTP molecule. When this GTP is made, we consider this substrate level phosphorylation because it's made from GDP plus an inorganic phosphate, and it can also be used as an energy molecule. So remember, those nucleotides are equivalent in terms of energy, so it could be used in reactions, or it can pass its phosphate onto other diphosphonucleotides. The net reaction will require 2 water molecules as substrates, and then the 4 reduced electron carriers, 3 NADHs and 1 FADH2, will then be used in oxidative phosphorylation to make more energy. Succinate was that symmetric molecule, and the enzyme that uses it will actually make the FADH2. And this is the one enzyme that's actually going to be membrane bound and links the citric acid cycle to oxidative phosphorylation. As soon as succinate dehydrogenase catalyzes the oxidation of succinate to fumarate, it contains a covalently bound FAD cofactor that will be reduced to FADH2. Now, the electrons that are found in FADH2 will immediately go into the electron transport chain. So on the right, I'm just showing you the structure for this enzyme, and it is membrane bound. So up here would be the inner mitochondrial membrane, while this domain down here at the bottom will be found in the mitochondrial matrix. And this is where that succinate is going to get oxidized to fumarate. The FAD cofactor will also get reduced, and we'll see that the electrons will be passed onto a molecule called coenzyme Q, and that's going to help pass electrons through to the electron transport chain. So succinate dehydrogenase has this FAD or FADH2 cofactor, and it actually goes by the name of complex 2, which is how we sort of refer to it in the electron transport chain. But these aren't distinct molecules. It's one protein, a huge complex of polypeptide chains and cofactors that actually participates in 2 different pathways. Okay. So this step, along with the production of NADH and the other redox reactions, are really important for energy production in oxidative phosphorylation. And that's all by linking the citric acid cycle to the electron transport chain. We looked at the thermodynamics of glycolysis, which was overall favorable under standard and cellular conditions. And the citric acid cycle also has reactions that are highly favorable under standard and cellular conditions. So this table is just showing you the 8 reactions, okay? And again, you don't need to memorize them individually. The details are just provided in case you're interested, along with the enzyme names that help speed up these reactions, okay? And those enzyme names should really help you understand and figure out what they're doing. Okay, so obviously NAD dehydrogenases, those are going to be important for those redox reactions where we're producing those high energy intermediates. We can see that under standard conditions, some of the reactions have negative delta G0 primes, while others are unfavorable, especially this last one that's catalyzed by malate dehydrogenase, and that's the one that's regenerating oxaloacetate. Under cellular conditions, we again see differences in the free energy changes based on the cellular concentrations of substrates and products. And what's really interesting is that under cellular conditions, the favorable reactions in the cycle are what's going to help drive the unfavorable reactions. So when you couple step 8 with step 1, the product of step 8, oxaloacetate, is going to be depleted in order to make citrate in step 1 in that favorable reaction. Now, it's these 3 reactions that have negative free energy changes that are going to be spontaneous and essentially irreversible. Okay, so these are the ones that are going to be regulated in order to slow down the cycle. The other 5 reactions can actually be reversible depending on the concentration of substrates and products. So the citric acid cycle really is the hub of metabolism in the mitochondria. It can use acetyl-CoA from either the breakdown of glucose via PDC or the breakdown of fatty acids that can also be used to make acetyl-CoA. And some of the amino acids can also get used to make acetyl-CoA or other citric acid cycle intermediates. Under standard conditions, the last step is unfavorable, but under cellular conditions, the overall pathway is favorable since we can couple step 8 with step 1 in the cycle, catalyzed by citrate synthase, using up that oxaloacetate and driving the malate dehydrogenase reaction in the forward direction. Under cellular conditions, there are 3 steps that are highly favorable, and these are going to be the regulated steps, slowing down the citric acid cycle when we have enough energy. And this will allow the acetyl-CoA to instead be used in fat synthesis or allow for the use of other citric acid cycle intermediates in other anabolic pathways. We can also produce some other of the non-essential amino acids from citric acid cycle intermediates. So even though we are drawing it as a cycle, some of the steps are reversible and you can have branching off to other pathways in order to synthesize other larger molecules. So really, as a cycle, the citric acid cycle is considered an amphibolic pathway because it does link anabolic and catabolic pathways together. The slide just summarizes the points of regulation. So at the top, I'm just showing you again the regulation of the pyruvate dehydrogenase complex, where ATP was inhibiting it and ADP was stimulating the complex. The products acetyl-CoA and NADH were also inhibiting the PDC, while the substrates pyruvate and calcium were stimulating it through those regulatory enzymes. Now, a similar trend can be seen for the first step of the citric acid cycle, where ATP and NADH, the high energy molecules, will inhibit this step in the pathway. And then you see ADP and pyruvate actually stimulating this first reaction. Steps 3 and 4 were those dehydrogenases that we looked at, and they both have large releases of free energy. And remember, this is also where we were seeing that oxidative decarboxylation happening. And they too are inhibited by the energy molecules. And they can also be stimulated by ADP and calcium, which we talked before and we saw was involved in muscle contraction and signaling. Okay, so when the cell needs energy, calcium's released, it can actually stimulate these enzymes. The product of the fourth reaction, succinyl-CoA, can also inhibit that reaction. So even though this might feel a bit overwhelming, trying to memorize exactly what molecules inhibits and stimulates an enzyme, try to understand the trends of which enzymes in a pathway will get regulated and how the high energy molecules can inhibit a catabolic pathway while low energy molecules will stimulate them to make more energy. The last thing I want you to think about is this cycle in terms of health and disease. Because it is so crucial in the breakdown of all fuel molecules, carbohydrates, fats, and amino acids, you really won't see a lot of mutations arising in the various enzymes. So which out of these enzymes do you think could have a mutation that affects its function but still allow a cell to survive without it? Think about what we've covered regarding the cycle and where things are made. Fumarase is actually the one enzyme that has mutations in it that will be the least lethal. Citrate synthase, or step 1, is very important because it's using the acetyl-CoA that's coming in from the breakdown of sugar, fats, and proteins, and you would be inhibiting the rest of the pathway. Step 3, or isocitrate dehydrogenase, was the first oxidative decarboxylation step, so we wouldn't be making that NADH for energy or any of the later steps. Step 6 is the enzyme that's also important for oxidative phosphorylation. So even though we'll still make two NADHs and a GTP, we'll be inhibiting the step in oxidative phosphorylation and we won't be able to make any ATP from FADH2. Fumarase is actually the one enzyme that does have mutations that are allowed, but it does result in rare diseases due to the buildup of its substrate, fumarate. When this step is inhibited, the pathway will slow down and excess fumarate will be detected in the urine and can lead to neurological symptoms. What it does do is convert this cycle into a linear pathway, and you can still make energy through these earlier steps, but there's actually another enzyme that can produce oxaloacetate from pyruvate. This will allow the cycle to run, just not as efficiently as normal. So there are some very important reactions and pathways that take place in the mitochondria in order to produce the molecules needed to make ATP. The citric acid cycle really is the key to metabolism, processing incoming metabolites from the breakdown of sugars, fats, and proteins in order to initially make energy. It'll combine acetyl-CoA with the final product, oxaloacetate, in the matrix, producing those reduced coenzymes that carry electrons to the electron transport chain for oxidative phosphorylation and ATP production. Mitochondrial bioenergetics will be inhibited when there is lots of energy and stimulated when the cell needs energy. So there really are three key steps in this pathway with large negative standard free energy changes that will be regulated. A single turn of the cycle will require two water molecules, generating those three NADHs, one FADH2, and a GTP. And remember, that's energetically equivalent to ATP, as well as the production of two carbon dioxide molecules. Be sure to review the main purpose of this pathway and the ways it's regulated before tackling the BBQs on Quercus.